Hello and a warm welcome from my side. My name is Katja Jakob and I am here together with Calliope Arcodi, who is a product marketing manager in life sciences at Zeiss Microscopy. And she will give you a little presentation about super resolution imaging on the LSM980 Airyscan to multiplex mode in a second. We are broadcasting from our headquarters in Jena, Germany, and we hope you enjoy this presentation. If you have questions, you can, or then we would recommend you stay tuned to this page because we will announce a Q&A session as well here so that you can participate there. And with this being said, I would say we are good to go. Calliope, I would hand over to you. Stage is yours.
Hello, hi, I am Calliope and I will talk to you in this webinar about the advancements in fast and gentle super resolution imaging introduced by the LSM980 Airyscan 2 multiplex mode. So in a nutshell, LSM980 is equipped with Airyscan 2 with multiplex mode and can help you image large volumes at high speeds, effectively moving us from an era of frames per second to an era of volumes per second. So here we can see an example of how we can use Airyscan 2 with multiplex mode to reveal the dynamics of chromosome movements during oocyte meiosis in starfish. As you can see on the left-hand side, we're looking at the movement of chromosomes in the oocyte, color-coded for depth over time. And on the right-hand side, we can appreciate a 3D rendering or projection of the volume of the sample color-coded for time. So these are observations that the research group was only able to do with the Airyscan 2 high resolution and sensitivity at high speed, allowed them to look in the chromosome diffusion and interaction with the actin and tubulin networks. So in this second example, you can see how LSM980 Airyscan 2 allows us to zoom in a brain explant and capture the cilia dynamics of the ependymal cells, imaging at 143 frames per second with the multiplex mode. This high temporal and spatial resolution allowed the researchers to correlate the information of the cilia movement with the fluid flow in the brain as a result they were not able to achieve with other methods, such as imaging with a resonance scanner. But before we look into more examples of how your work can be benefited with by our new LSM980, I'd like to talk to you about our overall motivation for developing it, which is a story dating a few years back. So back in 2014, we conducted a large survey asking researchers working with microscopy what were the key areas that needed improvement in imaging practices. The interesting part was that regardless of their field of interest or expertise, all researchers agreed that in fact they needed more speed, higher sensitivity, and enhanced resolution in their imaging. And that's exactly the inspiration behind Airyscan that expanded our understanding of what confocal microscopy is actually capable of. So as we can see here on the right, this is a traditional confocal image of a telomere replication study. You can see the different chromosomes, and the telomeres are the colorful dots at the end of the chromosomes. So the purpose of the study was to count the number of telomeres at the edge of the chromosomes. However, with traditional confocal microscopy images, that was not possible. Airyscan effectively doubles the resolution and increases the signal-to-noise ratio by 4 to 8 times, allowing us to image the underlying biology and clearly count the number of telomeres. Because sometimes these jumps in signal-to-noise ratio are the difference between achieving a result and simply not achieving it. So that was back in 2014, but the scientific world is ever changing and developing. And as quoted by Nature Methods, in the last few years, fundamental cell biology research has moved from studies in 2 dimensions, looking at cells on flat plastic plates, to more comprehensive tools and understanding how cells work in 3 dimensions, both singly and in connection to other cells. This statement was followed by the emergence of organoids as the method of the year 2017. So as the time goes by and we're transitioning from two-dimensional cells to more complex models such as organoids, tissue sections, whole organs, or organisms, the image acquisition must follow and allow to scale in size while keeping the most important structural information. So practically, looking at all these amazing different samples from organoids with volumes sizing up to 600 by 200 by 300 microns, to single neurons firing in zebrafish brains, to mitochondrial membranes, chromosomes, and even single microtubules in cardiomyocytes, we can see how diverse biology is. But once we scale up these samples against each other, we can understand that we need the microscopy abilities to scale up as well. So in essence, we need the performance of Airyscan in terms of sensitivity, resolution, and speed to scale up along with the model systems so that we can jump from frames per second to volumes per second that can efficiently cover an organoid or a whole animal in a reasonable time. So to summarize, working with organoids and whole model organisms as opposed to cells increases the complexity of the system, but also increases the physiological relevance of the results. However, traditional confocal microscopy has been centered around cultured cells in 2 dimensions and is not able to keep up with the modern, larger, and more demanding samples. Finally, another additional issue that researchers face when the model system is scaled up beyond cultured cells is the importance of maintaining the context of what you're actually looking at. So in the next few slides, we will look into what LSM980 and Airyscan 2 with multiplex mode can achieve and what tools we have available to help you set up your work and maintain the context when working with larger model systems. So let's first have a little glimpse into the beam path of the new LSM980 with Airyscan 2. So looking inside the scan head, we can see the laser that is passing through the twin gates and a series of optimized optics, reaching your sample, and then the emission light coming back, going through the optics, through the recycling loop that optimizes the light efficiency and mapped onto the 32-channel GASP detector. So let's have a closer look at the spectral capacity of the multi-channel quasar detector. So when you're imaging and the light, the signal reaches the quasar detector, depending on the labels that you're using, you can either define the emission detection bands with nanometer precision on the detector, or you can acquire overlapping labels in a single lambda scan with 34 channels and then separate them with linear unmixing. So as you probably know already, its fluorophore or absorbing dye has a unique spectral signature, its emission fingerprint, that can be determined independently and used to assign the proper contribution from that probe to every single pixel in the lambda scan. So the result of the linear unmixing is the generation of the distinct emission fingerprints for its fluorophore used in your specimen. So practically, sometimes it is inevitable that your sample will have fluorophores with emission spectral overlap, especially when you're working with transgenic lines where you have little to no control on what colors you want to use. So what you can do when your labels overlap is to scan your sample with a single lambda scan and let the linear unmixing to separate the dyes. It is quick, it is efficient, it is easy, and above all, it's gentle since it keeps the sample's exposure to laser light to a minimum. Let's have a look at the particularly difficult plant sample where the endoplasmic reticulum is labeled with CFP, actin is labeled by YFP, mitochondria in MT orange, and the endosome in FM464. While at the same time, since it's a live plant, it has strong chlorophyll autofluorescence. So in this application node, we utilized linear unmixing and emission fingerprinting to easily and quickly discriminate multiple labels with one single scan. So this allows you for a fast acquisition and dental imaging because we avoid sequential imaging of the different colors, allowing you to focus on what you're actually interested in, which in this case is the actin dynamics during the active movement of mitochondria and endoplasmic reticulum. So fixed samples also pose a problem of their own. They are very rich in the type of information we can extract from them, especially because we can label them via immunohistochemistry with various different fluorophores. However, imaging sequentially all these different fluorophores can create an experimental bottleneck in your work, resulting in longer acquisition times and sample bleaching. So with spectral imaging and unmixing, you have the opportunity to image up to 10 different fluorophores simultaneously, minimizing the acquisition time while maximizing the information collected. So in addition to the high-end hardware components, LSM980 is powered by ZenBlue. In one sentence, ZenBlue is a powerful platform containing multiple automatic tools that can help you do your work smarter, not harder. So to help you set up your imaging, ZenBlue brings to you the sample navigator that can help you find your sample and select your regions of interest within minutes. Used together with intelligent tiling that expands your field of view in a usable way, and smart setup, which is a clever, convenient way to set up your acquisition parameters for your imaging. And use all this together with the experiment designer that will allow you to set up heterogeneous experiments at the same time, variable parameters. So all these tools can significantly increase your productivity by decreasing the time spent setting up your experiments. But we didn't stop there. So you can use ZenConnect to bring all your data together in a comprehensive way and make sure nothing is ever misplaced. And 3DXL, our 3D visualization tool, which we bring to you in collaboration with Airyscan. You can use it to render 3D and 4D data sets that you have acquired with the fast LSM980, allowing you not only to visualize the structures, but also create impressive renderings and movies. And take your data analysis a step further and segment your data using Zen IntelliSys. So just use your own expertise to train the software on a few of your images, and then powerful deep learning algorithms will take over and do all the time-consuming segmentation steps of the hundreds of similar images. And if you want to go even further and continue your analysis, there is a great variety of tools in the ZenBlue analysis toolbox. So let's take a closer look at some of the key software features with an emphasis on how to increase the throughput of your work. So when you put your sample under the microscope, quite often the region of interest or even the whole sample is not within the field of view. A way to address this is to combine multiple fields of view, often referred to as tiling. But tiling can come with its own challenges. So specifically, very often, a slide is not 100% level, which means that some areas of your sample are going to be in focus, whereas some others are going to be completely out of focus. In order to avoid this, in the ZenBlue tiling menu can easily drop in support points. As you can see here, we have added 9 support points, which will be used by the software to map in order to mark focal positions across the tiled field of view, where the software is mapping the focus and making sure your sample is always in the correct position. So looking at an actual sample, this is how a tile scan with 1,334 tiles would look without a focus map. As you can see, parts of the image are in focus, whereas the majority of it is out of focus. And this is how the same sample looks with the use of a focus map, with all the neurons being in focus and ready for further imaging or straight analysis. So once you have your focus map, and your tile set up, and you have imaged a large area of your sample, one can consider that a problem of usually of using large areas is how one orient oneself in them. So Sample Navigator comes to address this issue, bringing you the equivalent of a GPS system for your samples. What do I mean by that? So assuming you are traveling in a foreign city or country, you wouldn't circle the streets trying to find the museum you're interested in seeing completely by chance. What you would do is that you would use your phone and pull out a map and try to orient yourself using that. And that's exactly what Sample Navigator does. So as you can see here, Sample Navigator is an automated workflow that effectively allows you to get an overview image of your sample with a few clicks. So first, you define the cover slip you're using, whether you want it image the whole size of the cover slip, and then you allow the software to define the focus. You can approve it by clicking Finish, and effectively, ZenBlue takes over and images your entire cover slip without the need for further input or interaction. So as you can see, the process is very quick. And effectively, you can image a 2 by 2 centimeter cover slip within 3 minutes. So once your cover slip is scanned, you can have a whole overview of your sample, and you can zoom in the different areas. It's important to note that sometimes the area you're interested in might not be exactly in the center of the cover slip, but might be further down. So another thing that's very interesting is that, as you can see, the quality of the image of the taken by the Sample Navigator is very high, and it allows you to clearly discriminate between different cells. And then you can change the magnification and image your different samples while you're maintaining the context of the greater area. And of course, Sample Navigator can be used with any sample, such as slides with multiple Drosophila melanogaster larvae, which would allow you to increase the number of individual samples imaged at a time. So to manage your data, ZenConnect has your back, allowing you to continue imaging at a later day at any system while maintaining all the previously acquired data in one place, allowing you to share your work with your group, your collaborator, or have it in a neat folder ready for a publication. Another great advantage of ZenConnect is that it allows you to organize your data even when they come from different microscopy systems while maintaining the context. So in this example, you can see an overview scan of a mouse brain, which can be used to map the super resolution data sets that were acquired from different regions, keeping your data organized and ensuring that nothing is ever misplaced or accidentally repeated. But as you know, biological samples can be as heterogeneous as life can be. And that's the reason that we have a huge variety of microscopes that can help you explore the area you're interested in. So starting from light microscopy, one can move to larger or denser structures observed with X-ray microscopy, or look in the super ultrafine structures by the use of electron microscopy and ion microscopy. Zeiss offers a huge breadth of microscopes to suit all of your needs. To sum up the first part of our presentation, till now we explored the exciting features of the new LSM980 and ZenBlue software. Now let's turn our attention to Airyscan 2 with Multiplex Mode and how it can help you take your research one step further. So to fully understand how Airyscan 2 Multiplex Mode works, we should first review how Airyscan works in comparison with traditional confocal microscopy. So in traditional confocal imaging, we are taking a laser and focusing it on a focal plane, moving the spot over the field of view to build an image a pixel at a time. So the fluorescence that is generated through this process is passing through the pinhole and is mapped onto a detector. However, Airyscan super resolution imaging works slightly different. So to begin with, we are also focusing a laser over a field of view and moving the diffraction limited spot to build an image a pixel at a time. But instead of focusing back the light through a pinhole and discarding precious light, the whole Airyscan detector is acting as a detector, a gas PMT array that sits on the confocal plane. So what does that practically mean, and what's the benefit of replacing a pinhole with the Airyscan detector? So as I mentioned before, in traditional confocal microscopy, the emitted light is passing through a pinhole that restricts the amount of light that goes through. So effectively, the smaller the pinhole, the better the optical sectioning ability of the system and the better the resolution achieved. However, when the pinhole gets smaller, more and more light gets discarded, resulting in images that might have theoretically a high resolution, but since there is almost no light coming through, this resolution cannot be realized. On the other hand, Airyscan detector is comprised of 32 detector elements that each and one of them represents a small 0.2 Airy unit pinhole, but the overall light collection efficiency is the equivalent of a larger pinhole on 1.25 Airy units. So as we scan the field of view, the Airyscan reads all the 32 elements at a time and forms a super resolution image, while simultaneously increasing the signal to noise ratio. So let's now have a look at exactly what closing the pinhole means practically by looking at an example. So looking at the diagrams, we can see that yes, indeed, the pinhole size does affect the slice thickness and resolution and light transmission. So the triangle symbol indicates the conventional confocal 180 unit pinhole. And as you can see, this convention allows us to collect enough light and have a good enough resolution to see the neuromuscular junction general structure in Drosophila, but we cannot fully resolve it. In order to improve the resolution, one could close the pinhole down to 0.2 Airy units represented by the hollow square. And this can actually improve the resolution, but would dramatically reduce the amount of light collected. So back to our neuromuscular junction example, what we can see is that you can still see the general structure of the neuromuscular junction and maybe start resolving some features, but the signal to noise ratio is too poor to realize the resolution improvement. Airyscan, on the other hand, brings you the best of both worlds. So as noted by the solid circle, it gives you an improvement in the resolution that you can get with a small 0.2 Airy unit pinhole paired with a light collection efficiency of working with a wider 1.25 Airy units pinhole. So looking in the image, we can clearly see the improvement in resolution and are now able to see the donut shape of the Drosophila neuromuscular junction. So Airyscan 2 Multiplex was designed to increase the imaging speed and field of view while maintaining the image quality. So as you can see, when we image with the Multiplex 4Y mode, effectively from a single diffraction limited spot, we can collect enough information that is enough to reform not 1, but 4 pixels at a time while maintaining super resolution and high signal to noise. So in essence, we can reconstitute 4 super resolution pixels from a single light limited spot, while you maintain high signal to noise and super resolution. So let's put Airyscan to multiplex in practice and see how this parallelization can be used. So seeing here an image of some cells taken with Airyscan 2 in super resolution mode. And here we can see an image taken with Airyscan 2 in multiplex 4Y mode, taken within the same amount of time. So as you can see, the field of view is significantly larger while the image quality is maintained. But we didn't stop there. we took it one step further in Multiplex 8Y mode, where the illumination beam itself is shaped along the y-axis, which allows us to reconstitute not 1, but 8 pixels or 8 lines at a time. This also allows you if you want to sample for confocal resolution or super resolution, depending on your experimental needs, while imaging at high speeds. So to look at how Multiplex 8Y mode of Airyscan 2 can help us push the boundaries of what's visible, let's first have a look at how the previous module of Airyscan, Airyscan Fast, allowed the researchers to resolve the structures. So as you can see here, these are some microtubules, and we can see with imaging at very high frame rates how the microtubules are buckling and bearing the load in cardiomyocyte contractions, an observation made possible only by Airyscan and not other technologies such as resonance scanners. However, if we now put what we were able to do with Multiplex 8Y mode compared to the fast mode, we can see how that we now have the ability to maintain the high temporal frame rate and the high spatial resolution and high signal to noise ratio, but over a much larger field of view, again providing greater context. Additionally, you can use this parallelization to increase the number of labels used, as the researchers did in this example, where they combined the microtubule labeling with the nuclear labeling, effectively allowing them to study the chromosomal expression, how chromosomal expression is affected by microtubule buckling. So to put the Airyscan advancements in the general context, and see how it compares with other technologies that increase your resolution or your speed, let's have a look at some COS7 cells, where the mitochondrial membrane is labeled in green and the actin is labeled in red. So a pretty modest acquisition volume of 72 by 72 by 6.7 microns. If you use a confocal imaging optimized for deconvolution, following the preset parameters for pinhole adjustment and averaging takes roughly 13 minutes. To improve the resolution of this image, you can apply adaptive automatic deconvolution with decision masks. And as you can see, the result of it is a better looking image, especially if we look in a specific cell zoomed in. So, however, if 30 minutes of acquisition time was not quick enough for your experiment, or if that would slow down your data collection far too much and is not tolerable, then you can couple the convolution with imaging with a resonance scanner. Resonance scanner moves the diffraction limited spot over the field of view much faster. And as you can see, it can bring down the acquisition time to 4.3 minutes for an even bigger field of view. However, the biggest drawback of using a resonance scanner is the fact that the pixel dwell time is decreased significantly, which results in a decrease in the signal to noise ratio and loss of resolution. So due to the poor signal to noise ratio, the convolution cannot necessarily improve the quality of the image in any significant manner. So if we look now at the same cells imaged with the same power using Airyscan 2 multiplex 4Y mode, sampled for super resolution, we can image a large 135 by 135 by 6.7 microns volume in 1.5 minutes, constituting a 20 times faster acquisition time for over a 3.5 times larger field of view. And most importantly, Airyscan 2 multiplex image clearly outperforms the adapted deconvolution in the resolution achieved, especially once we looked closer at a cell. So as you can see, the Airyscan image on the left, we can fully resolve the structure of the mitochondrial membrane labeled by top 20. whereas the image on the right that took 20 times longer to acquire, still the mitochondrial membrane is not clearly reconstituted. And if you would like the Airyscan image to be even better, you can even double the acquisition time from 1.5 minutes to 3 minutes and still have 25 minutes to spare, focusing on what actually matters the most. So the bottom line is that Airyscan Multiplex 4Y mode gives you a 70 times increase in the volumes images per second while retaining all the spatial information. So the confocal application we looked before used an objective lens of 63 times, which might be a magnification that is uncalled for your experiment. So this example was imaged with a 20 times objective, and as you can see, for a 258 by 258 by 34 microns acquisition volume, the speed of imaging was 61, the time of imaging was 61 minutes. So application of adaptive deconvolution with decision masks can once again help us improve the image. However, when we image the same sample with Airyscan 2, we can see that we can have the larger area image with only 4 minutes, while we are having 2 channels, 3.3 times larger volume, and we are 15 times faster in our imaging. And most importantly, when we zoom in, we can see that we can clearly visualize the dendritic spines when imaging with Airyscan 2, but not in the deconvolved confocal image. Effectively, Airyscan multiple 8Y results in a 50-fold increase in volume per second of imaging while retaining all the spatial information and allowing you to extract more data out of your sample. So similarly to what discussed before, if we look at the same sample using a confocal with a resonance scanner sampled for deconvolution, you can improve the acquisition speed while still not reaching the same speed as Airyscan multiplex, but it comes at the expense of structural information being extracted from your image. So as you can see clearly when we zoom in, all the ultrastructure of the neuronal spines is completely lost due to the resonance scanner imaging. So practically, multiplex mode delivers usable speed in your imaging. So as you saw in the previous slides, LSM980 with Airyscan 2 and specifically multiplex mode is able to support you as you move from 2 to 3-dimensional imaging without compromising. A parallel problem that researchers often have in large samples has to do with maintaining the greater context of where your structures are, as well as looking at bringing together the data collected by different people within the same group or collaborative groups that might or might not have overlapped in the timeline. So this issue is addressed by ZenConnect, which, as discussed before, brings together the different imaging modalities to a unified space. So having a look at a real example of scientific research, we can see how the researcher group used the multiplex mode in combination with ZenConnect to piece together the lab group's work from 2016 and bring it into the context with work that they did in 2019, leveraging ZenConnect to correlate the data collected at different time points and by different users. So in this case, the scientific question the research group was investigating was how the cilia-based flow networks in the brain work, and especially how the fluid moves in the brain based on the cilia dynamics. So what they did is that they created these amazing flow maps for the different areas of the brain ventricles, and they profiled in order to profile how the cilia move in the different chambers. So to create the flow maps, the movement of fluorescent beads was tracked using a stereoscope, and this experiment was done back in 2016. So what we were able to do in 2019 was to image a new brain explant and map onto the explant the flow maps that were created years before using the ZenConnect overlay functionality. So taking this a step further, the researchers were able to also map on the explant anatomy and the flow map the actual cilia movement imaged via Airyscan 2 multiplex at high frame rates. So in this case, you're looking at how the cilia are moving in chamber 7 of the mouse brain. And what was actually very interesting was the observation that cilia in different cells within the same chamber, they are not beating in a synchronous fashion. So this behavior was not apparent before and became visible only with Airyscan 2 multiplex mode. So looking in other areas of the brain, in this case chamber 6, at an even higher frame rate of 143 frames per second, the researchers were able to discover that the cilia movement asynchrony was also present within single cells. Once again, an observation made possible only by Airyscan multiplex mode. A different postdoc in the group was interested in the orientation of the basal bodies and the basal feet and how this affects the cilia beating direction. So in this case, you're looking at a fixed and labeled sample that presented a challenge of its own. So the researchers needed a super resolution image over a large volume, which traditionally took them 10 hours to do such an acquisition. So using the multiplex mode, we were able to cut this time in half, down to 5 hours, while maintaining the super resolution quality of the data. And when we look closer to the data acquired with an Airyscan 2 multiplex 8Y mode compared to a traditional confocal, we can see that the quality and the resolution of the image is actually higher. And it allowed the researchers to clearly visualize the polarity of the cilia, which dictates the beating directionality and therefore the fluid flow. So collectively, ZenConnect allowed us not only to bring together work done in 2016 and correlate it seamlessly with work carried out on 2019, but it also allowed us to take data from different users and other systems and projects and bring them together in a unified space. All this while maintaining the general context. So to summarize everything I discussed today, in one sentence, LSM980 with Airyscan 2 is your next generation confocal for fast and gentle multiplex imaging. So it gives you a great flexibility to set up your experiments using any of the software workflows and image with the highest speed and sensitivity. Multiplex mode with Airyscan 2 allows you to pair super resolution with usable speed while maintaining a large field of view. So thank you very much for your attention, and we're happy to answer your questions.
Thank you so much for this very interesting presentation, Calliope. I think people have enjoyed it, and as said at the beginning, stay tuned to this page and watch the next sessions so that you don't miss the Q&A session. Thank you so much to all of you who paid attention, and take very good care of yourself. Stay healthy. All the best from us, from Zeiss. Bye-bye.